All right, my name is Iniobong Afia, Chief Science Officer at Cannabis. Today we'll be talking a little bit about DCC Standardization and how to prepare for it. So this is really tailored towards analytical labs, but it does affect everyone here. Perfect. So back in October of 2021, California Senate passed SB 544 that was essentially about standardization, right? So let me see, my eyesight's pretty good. So pretty much what SB 544 was meant to implement provisions of the Marijuana Act, right, that required the DCC on or before January 1st of next year to implement some test methods, standardized test methods that the labs can use in order to test the potency of marijuana. It was supposed to be one or more, and it can be developed through a reference test lab. Again, we'll talk about what that really means. In the bill, there's 6 cannabinoids listed: THC, CH, CBD, CBG, CBDA, CBN. Can you hear me? Oh, okay, I should probably stick around here, right? I like to wander a little bit. So it talks about 6 cannabinoids. The concern for some of folks is, what does this really mean? What is standardization? And the fact that the bill only lists 6 cannabinoids, what does that mean? Are we going to be restricted to only testing 6 cannabinoids? And question is, will they really involve reference labs or the current California testing labs? As we know, there are over 46, I believe, or more of them currently. Also, there are lots of questions. Will it involve some NIST studies? I think a lot of labs here, if labs here, you'll know that there were a lot of studies performed through NIST, about 3 of them right now. And then the question can be asked, is standardization really a fool's errand, given the inherent variability of testing or cannabinoids in the plant? We know that your potency can differ from different flowers, from different parts of the plant, from the top, larger buds, to the bottom. So it's really, what are we standardizing? Will it stifle innovation? Right, it's a free market, and currently, each lab validates and tests its own test method for potency. Just in case anybody asks what is a test method, right? When you take your sample to a lab, they stick it on an instrument, usually an HPLC. For them to be able to run it, they have to develop and test a method and validate a method, right? So that's a test method I'm talking about. The method that they use to essentially provide your THC content. In that validation, they essentially are able to address certain things, certain parameters that the DCC set forth, okay? So one, we need clarification of that bill language. What exactly is to be standardized? DCC says you have to validate your method. You're validating, looking for accuracy, linearity, range, reproducibility, robustness, LOD, LOQs, which are a big topic, and specificity. What exactly is to be standardized? Let's talk about, is it the instrument method? As I just mentioned, the instrument method essentially is what is run on the instrument you're generally using, which is an HPLC, right? Do you standardize the mobile phases that are being used, the detector type that are being used? And you can ask me later if you have questions about those, what those really mean. Even the column that's being used. Very important, the sample prep method. Before that sample even gets on an instrument, how do you prepare it? What do you dissolve it in? What solvents do you use? And of course, the homogenization. Probably most importantly, the homogenization. How do you really homogenize that sample you're using, that you're about to test? And this is not just about flour, even the oils. And homogenization essentially means mixing it. I mean, I don't think I need to explain homogenization, but what are you using? A cryomill, as in terms of liquid nitrogen? Are you simply blending the products? What about the matrix considerations, right? You have gummies. I mean, cannabis is one of those, the cannabis industry is one of those industries where you can essentially test anything in the lab. I've seen so much that, every day I cease to be amazed. In terms of, I've tested toothpicks, candles, even whole mattresses, right? Folks are making mattresses infused with CBD. How useful that is, I don't know, but it's pretty exciting and pretty challenging to test. So the sample matrix considerations, right? Essentially, I just talked about that. All this myriad of products that are infused with cannabinoids, how do you really standardize testing that, or is that what we need to focus on in terms of standardization rather than the instrument test method? There's a thought about the use of compendial methods. We know folks like AOAC, ASTM, USP are working on, or do have currently standardized test methods. Should we follow one of those? Should the DCC think about going through one of those rather than developing a new test method that is required of the lab? This question is, does it create monopolies for instrument or consumable manufacturers, right? Valid concern. Generally, when you're creating a test method, I think when you're standardizing it, there's the general thought that you are not designating specific vendor part numbers, right? Let's say, for instance, a column, you wouldn't say, that's the test method must only use a Restec C18 Raptor column, right? Generally, there's nomenclature like a USP L1 column that designates a C18 column. I assume things like that will be done. Let's talk about what happens when there is no standardization. This is really why this is important. It's literally the slippery slope of lab shopping, which I'll show some data about. As I noted, what happens when a lot of things are happening around lab shopping? So what this is a client has a batch of flower, and obviously they want to be able to sell it for the highest value that they can sell it. So you have 48 or so labs in California, and you're going to use as many of them as possible to see how they compare, who's giving you the appropriate or the desired value, okay? Now, I'm not accusing anybody of doing anything unscrupulous here, but you're going to want to get your best result. So you're taking it to different labs, and they're giving you different numbers, and you generally will go with the highest one. And of course, this is not done on a compliance COA because you can only run that one time. So you get an R&D test, and then you like them, you rerun it as a compliance. When that happens, essentially, the consumer loses, right? Again, not necessarily that all labs are falsifying results, but if they happen to do that, you're giving a falsely high potency value, for instance, and the consumer loses because they go to a dispensary and they're paying much higher for a product that's may not necessarily be the number that is stated that it is. Or even more importantly, false negative results, right? So we're only focusing on potency, but think about the contaminants. You may be paying for something that should have failed for mold and yeast or aspergillus, right? Then we get into health concerns and the general public. The buyers and distributors lose because they may pay more for potentially inflated results. I mean, I don't know how many times I've seen this where, distributor buys batch of product from someone, right, and retests at my lab, and the value is drastically different, generally lower. And they would ask, well, what happened here? Of course, sometimes it's higher, but we'll get into why that may be the case. So that's a loss for them. Regulators lose control as you have labs just giving all sorts of numbers. And of course, arguments can be made around why these are different, especially when you're talking about flour. But when we're talking about a more homogenized matrix like an oil, a vape product, it should be a little more stable in terms of the testing values. And of course, the labs lose, right? So with inflation happening, it's funny. We're pretty much like the only industry where everything's going up, but our prices are going down because of the competition fighting amongst ourselves to, get clientele. So why is that? You go to your vegetables at the supermarket are going up, but yet our test costs are going down, and the consumables that we're buying are going up. So how do you keep up with consumables prices going up while your test costs going down? This explains why, labs are going out of business. Employees end up losing when this happens, right? Labs shut down and they end up on maybe unemployment. Taxpayers lose. Employment increases. Just want to note the timer is not going, so in case I do go over my time, you'll let me know. Let's look at some comparative forum data. There are plenty of data around that we've gathered, but I chose to sort of go off of this one. It's a nice set, just to give you a little glimpse into what's happening. Even the most recent NIST results that came out, the variability amongst it, it's really interesting. So this is comparative data from a client that sent samples to labs, about 8 labs, right? They did it 2 ways. It was a well-done study where you have a nice homogenized set of flour in this case, and when I say clean, it's generally there were no contaminants, right? We're gonna focus on potency on this one. So they send this clean homogenized flour, essentially ground up finely, to about 8 labs. You can kind of see, starting with terps, just look at the relative standard deviation. Obviously, there was one lab that had a terp content of 3.78%, which is quite rare in flour. It's an outlier, kind of sticks out. Why is that? On a ground flour that was sent to you, you still got 3.78%. I believe that result is false. You could see the congruency amongst the other labs' data. And total THC, about 21 or so percentage. Moisture was relatively stable, right? I like to go for under 10% RSD, but that wasn't the case here. And comparatively, this was a whole set of flour. So one was ground up, and the other was not. Whole buds were sent, and it was determined, or it was set that the bud sizes were generally about the same, right? So you can kind of discount the thought that, well, it could have been different bud sizes and whatnot. So notice what happens here. Notice the increase in total THC, the mean value of the same cultivar, right? From 21.6 to about 28.5. That's quite a jump from the same flour, right? Same product. Moisture stayed relatively the same. Total terps increased a little bit, relatively the same, okay? Notice how there was about a 32% increase in that mean potency value, but specifically lab 7 had a 13-point jump, right? That's a 59% increase in their total THC from the flour that was homogenized to one that was not of the same cultivar. That's a huge jump. So everybody generally went up when the flour was not homogenized and it was sent to them. So why is that? Interestingly, you'll see that even the non-homogenized flour, the RSD is actually lower than the homogenized one, right, around 11%. So everybody was around the same, but much higher, right? So you can see where things kind of get a little tricky here when you're doing, when labs are doing their own homogenization. And yes, you can note that homogenization is usually the key here to these numbers. Really interesting. Now you think as a distributor, you know, you get a lot more money for second data set, right? Let's look at contaminants this time. So the same vendor or client sent the same thing again, focusing on contaminants. This time she spiked it with aspergillus, right? For the same lab sets to do the same assay. Contaminated homogenized flour. Let's not focus on the potency, but you could see terps again, kind of funky all over the place, moisture. And now the labs knew a little bit more what was happening, right? Because it's coming from the same vendor, and you can see that the potency is generally higher when they know what the client expects, right? But look how many labs sort of failed, or I should say were accurate for detecting the aspergillus, which was well done again. This was well spiked, and it was made homogeneously. It was sent in by a microbiologist, so she knew what she was doing. There was a 62% accuracy in detecting aspergillus, okay? Of course, you'll see what's happening here. Now, when you look at a 62% accuracy in detecting aspergillus, that's concerning, right? So product that's out there, does it have the same rate of accuracy in detecting aspergillus? When the flour was not homogenized, guess what? Only 2 labs got it right, right? Again, it was well done, but this time it was not ground up flour, it was not homogenized. And keep in mind that how it would normally be sent to the lab would be non-homogenized, you know, whole bud. So now only 2 labs got it right. Really, really interesting and really, really scary. So forget about the potency lab shopping. Now we talk about public health, right? Public safety and contaminants. It's important to note that some of these labs, I think at least one, use a plate for aspergillus. There's a whole lot I could go down a rabbit hole around why that wasn't probably appropriate, but that was done. So you may note that one lab noted 2 species. Of course, there could be some cross-hybridization things happening, but the point is that they detected and failed the sample. But only 2 labs got it right. And of course, my lab's one of those. So is there a solution to all this? Well, yeah, there are solutions. We need to increase oversight of cannabis testing labs and their methods. Okay. I implore DCC to really step up their game in the oversight, especially in knowing exactly what to look for when they're auditing laboratories and the test methods they're using. Should perform unscheduled blind round robins. Right, a lot of times, you'll note that if a state or some kind of regulator wants to send a round robin through a lab, everybody knows what's happening. Generally, the numbers will be congruent. They'll look good. Be like, hey, what's the problem? Everything's good, right? But if you do on schedule where they don't know that it's sent by the state or whatnot, you'll see maybe a little bit of truth around that. I know all labs are required to perform proficiency testing, and they all do, and they all pass. Otherwise, they wouldn't keep their licenses or be able to test for those. So, of course, when you are taking a proficiency test, you know exactly what you're doing, and you're going to do things appropriately. There's thought of maybe the producers and cultivators should test in-house. Okay? Obviously, as a lab, I don't necessarily want that, but I don't necessarily mind that. But of course, there's a lot to be considered when you're trying to have them test in-house and knowing what to be expected before they send it out. So if a cultivator or distributor says, why is this 30%? If they were testing in-house and they already know that's not 30%, they wouldn't blame the lab for not reaching that, right? They would at least know what they're starting out with. Importantly is to solidify the Category 1 pesticide limits of detection. So right now, the state essentially allows labs to set their own LODs, especially for Category 1, based on their method and instrumentation. There's a cap, right, 0.1 ppm, but the labs are able to push that limit. We know the instruments can detect a lot lower than that. It's interesting to see from 2018, starting out here in California, the labs, their LODs, everybody was going for as low as possible, right? We want to be able to catch everything. As the years go by, you keep on seeing people's COAs creeping up. It's like, oh, we used to go at 0.001 to 0.008, and it just keeps creeping up, and now everybody's pushing 0.1. Well, hey, the state allows it, right? Why not? Why not take advantage of it if you can push the limit of detection, essentially not failing as many people for those Category 1 contaminants. And of course, those Category 1 contaminants are ones that you don't want present. Essentially, they're banned, right? But there's a residue limit that is expected for labs to be able to reach. You could place limits on THC content, right? So recent Mississippi rules is now placing limits on, well, if it sticks, I think it's the initial, they're making flowers fail at 30%. So it's like, why? Well, there's thought about how much THC can the flower really produce biologically? Can it really make 30% of its mass is only THC? What about all the other compounds? And when we're getting above 30%, is that true? Of course, there's considerations like we're testing by dry weight, meaning if you test flower, you test as is, so you get a 20% flower, but it's 10%, you dry it down and it's really 10% moisture. So essentially adding, it's not apples for apples, but essentially you're adding 10% more to that final number, right? So 20% becomes like a 22 or so because you have to test by dry weight. So for instance, if you're in Mississippi, well, forget about growing anything good, right? If you're at, 27 30, you're off, and they're adding in all the THC variants into the total THC calculation. But there's thought, should we place limits on that? They're also doing 60% for tinctures and oils and concentrates. Should that be the case? I'm not saying that this should be the case at all, but it's to be considered. And of course, eventually there'll be federal oversight, and I think that will really mitigate some of this unscrupulous activities that happens when you have federal oversight of these products. I've been in the pharmaceutical industry for years, and then I went to nutraceutical and finally cannabis. It's kind of interesting to see the shift. Initially, when I came into the cannabis industry, analytical side, I was coming in with the pharmaceutical mindset of really strict regulations and trying to apply that, but that didn't really apply in this industry. I had to step back and relax myself and go with the actual rules, right? I can't implement rules that are not being regulated by the regulatory bodies. But when there's federal oversight, you better believe there's going to be a lot stricter rules around this, and all these, lab shopping will essentially cease to be. There will always be folks out there that can do what they want, but I believe it'll cease to be. So how can labs really prepare for standardization? Luckily, SB 544 now allows DCC authority to set regulations for the list of contaminants and the respective action levels. So as much as we would like them to change things, some things cannot be changed that easily because you have to go through the regulatory red tape. Everybody has signed off, the governor, all of the above, right? So now that the Department of Cannabis Control will have that authority, they'll be able to hopefully make effective change a lot more quickly as to the contaminant list. Right, I know there'll be a talk here about increasing those contaminant lists. Man, where were all you guys before? So they'll be able to set that contaminant list, and it'll make things a little bit easier. You'll be able to speak directly to them, and hopefully we'll be able to see effective change when they're able to have that power to do this. Well, you should ensure that your instruments meet the sensitivity requirements that will probably change, right? So labs set up with different instruments. I was speaking of somebody using plates for aspergillus, Hopefully that will cease to exist because unless you have the appropriate staff and the appropriate methodologies to really speciate aspergillus on plates, you probably shouldn't be using it. There should be molecular methods. You should start validating to compendial methods. AOAC, SMPR, ASCM, USP, start validating to those. Make sure your validation is robust so that whatever the state does come up with, you'll probably meet it to begin with, right? It's interesting because I doubt the state will really force anybody into a method, but again, just speculating. We don't know. We're still learning what they're gonna do, or we're waiting to know what exactly they will do. You should run proficiency tests in the cannabis matrix. This is important for the proficiency PT providers. The industry has really grown. Initially, everything used to be insolvent. Yeah, I can take anything insolvent, shoot it, and it looks great forever until you run in matrix. It's funny, the initial data that used to come from some of the industry folks, it was all insolvent. And then you'd run it day to day in your instrument, on your column, and find out it's not robust, it doesn't last, because this wasn't done in matrix. You have to do this in matrix. I can't say enough how complicated and complex the cannabis matrix is. It throws a lot of curveballs when you're trying to validate in matrix. So to make sure that you're doing things right, you should run your proficiency tests in matrix. It means it should be supplied in a cannabis matrix, or at least a cannabis similar matrix. That way you know that your methods are robust and you're actually getting accurate values. You should evaluate your homogenization methods for consistency. I got a key point here for folks who were taking notes. Yeah, homogenization is key to some of these numbers. So when you're looking at gummies and you get, 10 packs of 20 count gummies, so you have a couple hundred of them and you're supposed to homogenize that. How do you really do that? Well, the rules kind of leave it open, right? Some people say, open all the bags and throw them together, shake them up, that's homogenization. Others melt it down, right? That's homogenized. Others cryo-grinded with liquid nitrogen to fine powder. That's homogenized. But this needs to be standardized in order to be able to effectively make sure you're consistent. evaluate the CRM vendors, right, certified reference material vendors, and stability of mixed components, I can't say many times I've been able to kind of prove CRM vendors wrong that their mixture was not stable, right? Not trying to boast out there, but there are, when you're trying to put all these components into one vial and say, oh, this will last for 2 years, well, generally, they found that it doesn't, right? And the industry has matured, and they found they need to separate some of these compounds as they do not mix well and remain stable in solution. Be ready to run and maintain multiple methods and instrument methods, because chances are a lot of you are already doing it, right? A lot of labs are already running and maintaining multiple methods. So just, if the DCC does require that you will be using that, you're probably going to have to run multiple methods. So with that, right on time, I'd like to take some questions. Yes.
Sorry, I'm short here.
Yeah.
Thank you very much, and thank you for the hard work that you do with DCC. I have a couple of questions for you. One is, my understanding is that DCC requires laboratories to be ISO accredited. Is that correct?
Yes.
Okay. How familiar is DCC with ISO 17025?
Well, honestly, a lot of the regulators, the members of the DCC did come from like environmental side and pharmaceuticals, so they should be very familiar with it, and they do require that accreditation. But interestingly, whenever an audit happens, usually they're not touching on that, right? So I would expect them to touch more on that side of that accreditation, of the required accreditation.
Okay. You made a couple of comments that I'm hoping you can clarify for me. One is, you said all methods need to be validated. First, you said that all in-house methods for potency are in-house methods that the laboratory has to validate. And then later in your presentation, you mentioned that a standard test method could be used, a compendial method such as AOAC. Mm-hmm. Correct?
Yes.
Okay. So I'm wondering, and help me understand this, if a laboratory is using a standardized compendial method, let's say something from, I think you quoted USP or AOAC, mm-hmm. whatever, mm-hmm. Do you allow the laboratories in California to verify their ability to meet the standard compendial method requirements, or do you, which is required by ISO 17025, or do you require the laboratories to ignore that part of 17025 and require them to validate even a standardized compendial test method? That's part one of my question. And part two of my question is, if at some point they make a minor change to one of their methods, let's say they're moving from a 20 gram or 20 milligram sample size to a 18 milligram sample size, do you require them to go through the whole validation process all over again?
So I'm not the DCC, but very important, great question. For one, this really revolves around standard operating procedures, right? So if you're running a compendial method, you should at minimum verify that in your laboratory on your instrument. It's not required to do a full validation. Now, I say that because the DCC has done it several ways, where in the microbial section, they require full validation regardless, right? You bring it in-house, you do a full validation, but on the more chemical methods, it's been at minimum verification. This is important in your SOPs, because per ISO regulations, I mean, ISO is really about doing what you say you're doing, right? So if you write in your SOPs that I will only be verifying compendial methods for these certain parameters, you should do that. And that's really how it should be done. There would be really no need to revalidate in its entirety a compendial method, since a lot of that work was done. So no, yeah, it would be based on your SOP, and you should revalidate. You should do that based on your SOP. If you do change a parameter of the method, that's a compendial method, again, those parameters could be based on your SOPs, but we all know what the standard is. Yes, you should revalidate the entire thing. If the method calls for a certain column, a C18, 45 millimeter length or whatever, and you change it to, I don't know, hydrophilic column, that's a whole new validation because you're not following the compendial method.
One more question, if I may. And that is around your CRMs. So certified reference material, I believe those are accredited also to an ISO standard,
Yes.
right?
Yes.
Does the DCC require the laboratory to establish the shelf life of those standards once they've been cracked open?
So no, they don't, and that's a very important point. They really should, right? So the vendors, yeah, they do validate this, but they haven't done any shelf lives testing, at least long term anyway. I think they're starting to now, or some have already, but generally I don't see it, right? So they're not testing stability. A lot of times I've seen them do it in the process. They introduce a product out there. It's good when produced, and then they're doing the study concurrently. A few months later, you get this amendment of a COA. Oh, we found out that this is no longer this. So, you have to get the product out there at the same time.
So I think the difference is that the CRM comes with an expiration date on it.
It does.
Right? And it could be for 5 years or 2 years
Exactly.
or whatever. But that expiration date is null and voided as soon as
you open it. Right.
Vial is cracked open. So if I get a standard today and it expires in 5 years, let's say it expires in 2027, as soon as I open it, the CRM provider has absolutely no way to develop or know what that shelf life is.
Exactly. The onus
That ownership is on the laboratory, not on the vendor.
is on the lab, exactly. And some of these things that happen that you may suspect is unscrupulous is really just negligence, right? If you're calibrating and you have some old THC standard that's mixed and open, stored in your freezer, that THCA is converting to THC over time, and now you're inflating your THC value and deflating your THCA. You see how that can happen. So yes, the onus is on the lab to make sure that.
So that responsibility shifts to the lab, not on the CRM. Anyway.
Yes, it should be, but I do expect that the CRM vendor will at least make some sort of, I don't know, there bears some responsibility for at least limiting that, knowing that this will be open and used. I think it should be a shared responsibility, but that's just my opinion.
Thank you very much for your help.
Thank you.
Hi, Nick, thank you for the presentation. I had a question on the RPD that's allowed by the state. For every single method, it's plus or minus 30%.
Right.
At part per trillion levels, that's acceptable. At percent levels, it's a disaster. So anybody that's plus or minus 30% on their potency numbers is well within the regulations of the DCC. So the question is, if they are going to be coming up with certified or reference methods, could that be tightened to in any way, and what would you see as an acceptable RPD for potency?
Yeah, fantastic question, Art. I mean, the RPD is super wide, 30%. Of course, you don't know the expected results of the generally potency ones, right? And if it's in PT, PT are even wider, they're 40%. So you could pass them in, you could drive a truck through that. They will have to tighten that. In the lab, I expect around 10%, but really, I guess you work around measurement uncertainty. You have to know what your measurement uncertainty for your analytes are. But yeah, those do need to be tightened because you can't have 30% that wide and you can just, it could be anything and everybody will pass regardless, between 15 to 30% of a expected number. That still passes. So yes, I expect that'll be tightened as well. All right. Thank you very much, ladies and gentlemen.